Hi friends, we are going to see the past year NEET questions for the chapter Biotechnology Principles and Processes, and today in this video we will cover the next part, which is part 2. So let's begin with the first question here. Which one of the following represents a palindromic sequence sequence in DNA? So what is a palindrome first of all? How can you describe it? Palindrome, right? Palindrome is where I'm sorry, I didn't put in the solution. Okay, it's a word or a phrase or a sequence that reads the same backwards as forward. So even when you read it from the forward or when you read it from the back, reverse direction also, it should have the same set. Forward and the backward will be the same. So you can easily identify from this. The correct answer is option A. So if you read it the other way around, if you read it from the, see here, if you see here, GAATTC, GAATTC. So you read it other way around also, you get the same this thing. A palindromic sequence is a nucleic acid sequence of DNA or RNA that is the same whether read it from the 5' direction or the 3' direction. Okay, whatever be this thing, it's the same. So palindrome, the common meaning is this. The same thing follows here. Any way you read it, it will be the same. Therefore, the correct answer is option A. Next question here. Which one of the following, which one of the following technique made it possible to genetically engineer living organisms? Okay, so here you have 4 options, right? So what do you think will be the answer here? See, one technique has given you the process of inserting, deleting, making your own interest. You can create species which are resistant, which are deficient, so many things. So you have 4 options here: hybridization, X-ray diffraction. It is obviously not X-ray diffraction, not hybridization. The correct answer is recombinant DNA techniques. Recombinant DNA technology is a process which helps us to join together 2 DNA molecules from 2 different species that are inserted with the help of a host organism to produce a new genetic combination of the organism. Therefore, the correct answer is option A. Next question here. There is a restriction endonuclease called as EcoRI. What does CO part stand for? CO stands for the coli, because we have isolated from the organism E. coli. So that is why instead of mentioning the full organism name here, actual name is Escherichia coli. So we actually write it in the form of this form. So the CO represents the coli, the name of the organism. It's an endonuclease enzyme that is isolated from the strains of the Escherichia coli, and as a part of the restriction modification system, CO represents the coli. Very easy question, right? Next question here. Which one of the following is used as a vector for cloning genes into higher organisms? Okay, this is again an easy question for you. You have, obviously it is not Salmonella typhimurium, which causes typhoid, baculovirus, Rhizopus nigericans, and you have retrovirus. The correct answer is option D. Retrovirus has the ability to transform normal cells into cancerous cells. Hence, it can be used as a vector for cloning desirable genes into the animal cells. So because of this particular quality they are used, because we need to modify something, right? So this retrovirus has the ability to transform normal cells into cancerous cells. Therefore, the correct answer to this question is option D. Next question here. Which one of the following is a palindromic base sequence in DNA and can be easily cut about the middle by some particular restriction enzyme? Okay, again it's a palindromic sequence, so it's again option C here. Okay, so you read it any way around, it's going to be the same. I just now explained in the very first question of this video what is palindrome. You read it from A to B or B to C, it should be the same, right? So here if you see GAATTC, GAATTC. In the given question, only option C represents a palindromic sequence. Therefore, the correct answer is option C. Next question here. DNA or RNA segment tagged with a radioactive molecule is called as dash. I think this we already discussed in the part 1. By now you'd have guessed the answer. So when you have, see, certain questions will give you hints. They will give you actually like keywords which will help you easily identify the answer. If you remember, if you clearly watch the part 1, now you will be able to tell the answer. You have the keyword, which is radioactive molecule. The correct answer is probe. DNA or RNA segment that is tagged with a radioactive probe. So here you have the keyword. What's the keyword? Radioactive. It's radioactive molecule or a probe. They are used to detect the presence of the complementary sequences in the nucleic acid samples. So what is the main purpose of them? Is to identify. Right, that is the main purpose. So here probes are used for the identification and isolation of DNA or RNA. Therefore, the correct answer to this question is option B. Next question here. Restriction endonucleases are enzymes which make dash. Easy question because we already discussed about the activity of restriction endonuclease. Restriction endonucleases or enzymes, they actually help you cut. Right. So it makes a cut at specific position within a DNA molecule, and that site is called as a recognition nucleotide sequence. It will not cut anywhere and everywhere. That is not possible. It will only go for the recognition site and cut. Restriction endonucleases are enzymes that make a cut at the specific positions within the DNA molecule. They act as a molecule scissor. They recognize specific base sequence at palindrome sites in DNA duplex, and they cut these strands. So the duplexes can be recognized and be cut, that is eradicated from the sequence part. So it will not help, it will not lead you into any malfunctioning. Therefore, the correct answer is option A. Next question here. Stirred tank bioreactors have been designed for dash. Now, what do you mean by a stirred tank bioreactor? A stirred tank bioreactor is a cylindrical or a curved base. It's a curved base to facilitate the mixing of the reaction content. Okay. It has been designed for the availability of oxygen throughout the process. So why do you have a stirred tank bioreactor? Once you keep stirring, there's a lot of air circulation. So the oxygen requirement is found throughout the medium. It's found uniformly or equally. So this stirred tank bioreactors have been designed for the availability of oxygen throughout the process. That is why it will keep stirring throughout the process. The correct answer is option D. Next question here. Polyethylene glycol method is used for dash. Okay, it is obviously not biodiesel production, seedless food production, energy production from sewage, nothing like that. It is actually a gene transfer without a vector. Direct gene transfer is a transfer of naked DNA into the plant cells, but the presence of a rigid plant cell wall acts as a barrier to the uptake. Therefore, the protoplasts are the favored target for the direct gene transfer. So if you're going to do a direct gene transfer, sometimes the plant cell walls will reject it. Okay, so what do they use? Instead, they use a protoplast. Therefore, protoplasts are the favored target for the direct gene transfer. Polyethylene glycol mediated DNA uptake is a direct gene transfer method that utilizes the interaction between polyethylene glycol, naked DNA salts, and protoplast membrane to effect transport of the DNA into the cytoplasm. So when the cell wall becomes a barrier there, they have to use another medium. It's a direct gene transfer where you do not use any vectors. Therefore, polyethylene glycol method is used for the gene transfer without a vector. Okay. Next question here. Which one of the following is commonly used for transfer of foreign DNA into crop plants? Very easy question. What do you think will be the answer here? Yes, the right answer is Agrobacterium tumefaciens. It is the casual agent of the, what disease does it cause? What is it called as? Crown gall. You must have seen the images on net. They appear like huge lumps. Right, it's called the crown gall diseases, the formation of tumors in over 140 species of dicot. This disease is caused by the DNA plasmid, which is TI plasmid, and it's carried by the bacterium and transferred to the plant cells. TI plasmid is widely used in plant engineering as a vector in order to inject the gene in the host plant to form a transgenic plant. Therefore, the correct answer to this question is option B. Agrobacterium tumefaciens. Next question here. The linking of the antibiotic resistant gene with the plasmid vector becomes possible with dash. Okay, you are using a linking, and that too with an antibiotic resistant gene with a plasmid vector. So you need something for its attachment. So you do it with the help of, say, endonucleases, DNA polymerase, exonucleases, DNA ligase. DNA ligase is the enzyme that is able to join together two portions of the DNA and therefore plays an important role in the DNA repair mechanism. DNA ligase is also used in the recombinant DNA technology, as it ensures that the foreign DNA is bound to the plasmid into which it is incorporated. Apart from that, if you want to know anything about ligase, DNA, what is it called as? DNA ligase. See, a DNA ligase is a specific type of enzyme. It facilitates joining. Okay, so it plays an important role in repairing single-stranded breaks in duplex DNA in living organism, but some forms may be specifically repair double-stranded breaks also. So this is just an information that I would like to give here. Okay, so the main activity is to join. Okay, therefore the correct answer to this question is option A. Next question here. Gel electrophoresis is used for dash. I think you will know the answer of this because we discussed it in the part 1. We all know gel electrophoresis is separation method, right? Yeah, the correct answer is option B. Separation of DNA fragments according to their size and the charge. Charge they haven't mentioned here, but it's basically a separation that is based on two categories. One is size and the other is charge. Okay, gel electrophoresis is a technique which is of separation of DNA fragments according to their size. DNA is negatively charged, so in gel electrophoresis tank, which when electricity is passed, DNA moves towards a positive electrode. So once again, it is involved with charge also. Therefore, the correct answer is option B. Next question here. What is an antisense technology? This has a very big explanation to your right side. So we will read it and I'll explain it to you. Antisense RNA molecule whose base sequence is complementary to that of RNA subtranscript of a gene. That is the sense RNA. So when I say sense RNA, you can also talk about the messenger RNA. Apart from that, what else you can remember here is antisense. Basically, what is this antisense? In genetics, a sense strand or a coding strand is a segment with double-stranded DNA that carries a translatable code from 5' to 3' direction. Okay, now when the same thing, antisense strand of DNA or template strand which does not carry any translatable code. So this is the difference between the sense and antisense. Sense will carry the code while antisense will not carry any code. So you can say no code, no translatable code. Okay, so what is antisense technology? What is the answer here? The correct answer is option C. When a piece of RNA that is complementary in sequence is used to stop an expression of a specific gene. Since it carries no code, what activity will it perform? It will not perform any activity. Therefore, the correct answer is option C. Next question here. Chromosomes in a bacterial cell can be 1 to 3 in number and dash. Okay, so when we talk about a bacterial cell chromosome, how is it actually in appearance? It is circular. Prokaryotes, bacteria have only circular chromosomes. That is something that you should know, otherwise you will not be able to answer this. Bacteria belong to prokaryotes and their chromosomes are circular. So they are always circular. The correct answer is option B. Next question here. In bacteria, plasmid is dash. Yes, all of you will know what the answer is. Yes, it is option A, extrachromosomal material, and it's once again a circular plasmid. Plasmids are actually attached to the mesosomes of the bacteria. So in bacteria, plasmid is the extrachromosomal circular DNA material. Therefore, the correct answer is option A. We already discussed a lot about it in the part 1, so I haven't given much explanation. The question is direct and it's an easy question also. So with this, we complete the part 2 of biotechnology principles and processes. Hope to meet you in the next part of the same chapter. Thank you.